or good afternoon almost. My name is Ana Maria Blanco Martinez, as I was introduced, and I come from the Faculty of Medicine from the Federal University of Rio de Janeiro in Brazil. And I'm going to talk to you today about strategies to improve regeneration after peripheral nerve trauma. So we know since the beginning of the 20th century, from the work of Santiago Ramon y Cajal and Spanish neuroscientists, that the peripheral nerve system is able to regenerate. Cajal saw that after an incomplete lesion of a peripheral nerve, there was regeneration across the injury site. He also saw that after a complete lesion, not many fibers crossed this gap. Cajal even said that this ability to regenerate was due to the plastic capacity of Schwann cells, the cells that make myelin in the peripheral nervous system. And all of this is still true today. For those of you who are not acquainted with this subject, I brought this cartoon. And here is a normal neuron, the cell body, and the axon with its myelin. And when there is an injury here, the distal stump will become degenerated in a process known as Wallerian degeneration. So the axon and myelin become fragmented, and this is being phagocyted by Schwann cells that stay here in the distal stump, and also macrophages that come from the peripheral blood. So they will clean this way to allow regeneration. While this is happening here in the distal stump, the neural cell body starts to increase the amount of protein synthesis so as to start regeneration. So regenerating sprouts come from the proximal stumps, and some of these sprouts are able to reach the tissue, the target tissue. They will grow alongside the basal lamina of Schwann cells. And then these axons become remyelinated by these Schwann cells. And somehow the function is restored. However, despite of this intrinsic capacity, regenerative capacity of the peripheral nervous system, functional recovery does not always happen, particularly in men. So why is that? In such lesions, for example, in a proximal lesion here of the brachial plexus, if a lesion is here, it means that it's distant from the target tissue, the muscle in the hand, and this distance is about 80 centimeters. So taking in consideration that the growth rate of new axons is 1 millimeter per day, it means that it will take 800 days for these axons to reach the hand. So this is a long time. This is more than 2 years. What happens during these 800 days? It happens that the growth capacity of the cell body of the neurons will decrease over time. And also the plastic capacity of Schwann cells supporting regeneration will also decay over time. So at the end, not many fibers get into the tissue, target tissue. And so regeneration is not recovery, functional recovery is not total, and the person remains with functional deficits throughout their life. What can we do to overcome this limitation, at least experimentally? We can try to increase the number and velocity of regenerating nerve fibers. We can try to keep the regenerativity capacity of the cell body neurons for longer times. We can try to keep the trophic capacity of Schwann cells for a longer time as well, and we should avoid muscle atrophy as well. What surgeons meet in their clinic? So, when there is a lesion to a peripheral nerve, but there is no tissue loss, What the surgeon do is they approach the proximal and distal stump, and they suture it through the epineuron. However, if there is a gap, if there is tissue loss, this cannot be done without tension, further tension that would make another lesion. So what the surgeon does, and what is called the gold standard for bridging long nerve gaps, is what we call autologous nerve graft or autograft. And so the surgeon make another surgery to take a segment of a health nerve and use it to bridge the lesion site. Of course, this technique has many disadvantages. And one of these is that it needs a second operation to get the nerve. And of course, there is donor site morbidity because this nerve is gone, and the area innervated by it is no function anymore. Another technique that is being used by surgeons, neurosurgeons, is what called nerve transfer. This is used when the proximal stump of the nerve is not there anymore. When this very proximal, for example, the lesion is very proximal, the surgeon cannot find it. So he uses a health nerve. He takes some fascicles from this health nerve and bridge it, suture it to the distal stump of the injured nerve. The disadvantage of this technique is that it may result in loss of function of the donor nerve muscle, if it was a muscle nerve. Another technique that is also used is what we call the use of nerve conduits or tubulization. This technique is used in the clinical practice only for very thin nerves in the hand and for short gaps. It is said that a disadvantage of this technique is that it cannot be used for long nerve gaps until today. This can be changed in the future. The use of these conduits have been very explored in the experimental set. These tubes, they can be made from either synthetic or biological materials, but it is desirable that they present the following qualities. They should be biodegradable, and histocompatible. They should support suture. They should not collapse on the nerve that is growing inside. They should allow the entrance of nutrients, so they should be porous to allow the entrance of nutrients. They should be good substrates for axonal growth. And also, they can be used in association with the other strategies, such as cell therapy, gene therapy, and molecular therapy. We have been working with to obtain a porous tubes in our lab made from a polymer of lactic acid. And to make these tubes, first we do the membranes, because the external surface of the membrane has to be porous, for the reasons that I already said. And the internal surface, it should be less porous, so as to keep the regenerative good stuff inside the tube wall. And after that, we make the tube by this extrusion technique, and this is a nice technique because you can change the internal diameter of the tube and also the thickness of the tube wall according to the nerve that you want to bridge. We have evaluated these tubes by a scanning electron microscopy to see if they support the growth of cells. So we cultured Schwann cells and mesenchymal stem cells on top of them, and you can see that these cells attach to the surface and they make this long process. So they are nice for these cells. The surgical implantation of these tubes. Here is a mice sciatic nerve. Here's the nerve after being cut, and here's the nerve after being bridged by this tube. We have made morphological quantitative analysis to search for regeneration. And here is a cross-section, and the cross-section is in here in the sides because we leave a gap here to challenge regeneration. So in this area, there was nothing before. So everything that we find here is regenerated tissue. So we do this cross-section here, and here we have our PLA tube, And we compared it with a polycaprolactone tube, which is a PCL tube, which we used before and we knew it gave good results. So you can see that both tubes gave good regeneration of the nerve, and the nerve fibers look very healthy under the electron microscopy. Here we counted the number of myelinated fibers, and you can see is the total number of fibers, and you can see that our PLA group have given better results. So it was for the number of blood vessels. The fiber area was bigger in our group, and the myelin area was also thicker in the group that received the PLA tube. Functionally, to measure the function of the sciatic nerve, we use these paw prints. This is a paw print of a normal animal, and this is a paw print of an animal that had the sciatic nerve cut. And we measure the two spread and the print length, and we put this in this formula to get the sciatic functional index. And when we plot the results, you can see that the blue one is the PLA, gave better results. And the same for G-rate. G-rate is a measure that is obtained by dividing the internal diameter of the axon by the external diameter of the fiber. And for the sciatic nerve, the normal range of this G-rate is about 0.6 to 0.7. And most of the fibers fell into this range in case of the PLA. So functionally, they were even better than the PCL. We didn't expect it. We expected it to be as good as the other one, but it was better. Another strategy that we have been using is use the conduits plus cell therapy. And in this case, we use the mesenchymal stem cells and in a model of median nerve injury in mice. So the design was the same, but in this case, we introduced the cells here in this gap, the mesenchymal stem cells obtained from bone marrow. And here is the cross-section of the nerve, taken from here. This is a normal nerve, median nerve. Here is the nerve from the group that received the mesenchymal stem cells plus tube. And here is the nerve that received the tube plus the culture medium. It's our control. You can see that there was regeneration, but when we counted the number of myelinated nerve fiber, this number was higher, significantly higher, in the group that received the mesenchymal stem cells. And so was the number of blood vessels. We even measured the number of myelinated nerve fibers, and these are only counted, are only seen in ultrastructuring under the electron microscope, and when we counted them, they were higher in our MSC-treated group. We sorted to see if the cells that we inject were able to differentiate into other cells, and it was positive for Schwann cells. And you can see here is a DAPI. Here is GFP because the cells we injected were from a GFP positive animal, so we could follow them. Here is a X100, that is a marker for Schwann cells. And you can see that some injected cells presented the marker for Schwann cells. So at least some of them were able to to differentiate into Schwann cells. To measure the function of the median nerve, we use the grasping test. In this test, we put the animal on top of a grid, and this grid is placed on top of a balance. And we pull it gently, and when it leaves the grid, the force, the grasping force, is set here. And when we plot this result, we could see that our MSC-treated animals had a much better grasping test compared with our control. We also evaluated the degradation of the tube. In this case, we used another tube. It was a polycaprolactone tube, and it's also biodegradable, so we evaluated this by scanning electron microscopy. And you can see that by 12 weeks, the thickness, the wall of the tube is becoming degraded as we expected. We painted the nerve inside in yellow, gold, to see that the nerve is growing inside the tube wall nicely. This is one of our previous work in which was the first work, in fact, that we used stem cells from bone marrow in a collagen tube. And we did this scanning electron microscope to see how regeneration was. And you can see the tube here, the thickness of the tube. And inside the tubes, you can see these very nice nerve fibers growing in parallel with each other and very well aligned with blood vessels that looked very healthy. This was called our attention because we could not see this in our control group. So we asked ourselves at that time if the cells that we inject, the stem cells, were able to secrete factors, for example, vascular endothelial growth factors, that would allow regeneration as well. So we started another project in which we used the tubes plus gene therapy with VEGF in a sciatic nerve lesion model. So the design was very similar, but in this case, we used the gene, human VEGF 165, in the muscle below the nerve, because we didn't want to inject it in the nerve to make another lesion. And after that, we did electroporation to allow the entrance of the gene inside the muscle. And then we cut and tubulize the nerve. And you can see here that 1 week after injury, the amount of VEGF is much higher in comparison with the control that did not receive the gene. So morphologically, now you know how I do it, we can see that the VEGF-treated groups presented many regenerated nerve fibers. They looked very healthy under the electron microscope. And when we counted the total number of nerve fibers, they were much higher in the VEGF group. And so was, as we expected, the number of blood vessels. Function by sciatic functional index was also much better in the VEGF-treated group, 6 weeks after injury. We wanted to see if there was an increase in the neuronal survival, and this was responsible for the better regeneration. So we counted the number of sensory neurons in the dorsal root ganglia and the motor neurons in the spinal cord, in the anterior horn of the spinal cord. And yes, there was a much higher number of neurons in the dorsal root ganglion in our treated group, and so it was the number of motor neurons. So there was an increase in neuronal survival in our treated group. We started another, because these strategies, they don't make a full recovery. So we started another project in which we associated tubulization, cell therapy, and treadmill training exercise, also in a model of sciatic nerve injury. Here we have 4 groups, because now we have the control groups, the group of animals that just went to the treadmill training, the animals that received Schwann cells. In this case, the cell therapy was with Schwann cells, and treadmill training plus Schwann cells and plus tubulization. So we can see that the 3 treatments gave good results in terms of number of myelinated fibers, but only the group that received the 3 strategies had significantly better results. So it was the same for myelin area, and there was no difference among these 3 for axon area and fiber area. For functional analysis, the 3 groups for the SCFE, the 3 groups gave better results compared to the control group. But the one in blue here is the one that received the 3 strategies, and this was statistically better than the other 2 groups. We want to see if the transplanted Schwann cells were able to form myelin in the host tissue. And here is the same, DAPI, GFP, S100, and you can see that the cells that we injected maintained the marker S100. And then we did an immunogold electron microscopy to see if the cells that we injected made myelin in the host tissue. This technique is a little bit different, but here you have the axon, the myelin, and here is the cytoplasm of the Schwann cells. And you can see these black dots here. I hope you can see them. These black dots are nanogold particles associated with GFP time. So this is, means that the cell that we injected were able to form myelin in the host tissue. I'm finishing. We also searched to see if the cells that we injected were able to secrete neurotrophic factors, and we saw it in vitro and in vivo as well. And you can see that the group of animals that received the three strategies released a higher quantity of BDNF, NGF, NT3, and NT4. And that's it. Thank you very much for your attention. And